Conoce más del Instituto Carlos Slim de la Salud.
Our work is focused on identifying somatic genome alterations in breast cancer. Somatic alterations in the genome are the changes in genes that happen between normal cells and cancer cells. These changes in genes are important because in some cases they may represent targets for cancer therapy. In our study, we analyzed the genomes of 108 breast cancers. In some cases, we looked at the entire genomes. In some cases, we looked at what are called exomes, the sequences of the genes that code for proteins. We have analyzed huge amounts of data have reported these. I should comment that studies of this nature in cancer genomes were completely impossible as recently as 5 years ago and have been enabled by dramatic and revolutionary advances in DNA sequencing technology. And that our study and the other studies published together with ours in Nature are by far the largest genome studies of breast cancer ever conducted or reported. And I believe if you put them together, the largest studies of the genomes of any cancer ever reported. The major findings of our study were several newly mutated genes that had not been previously discovered. First, there were genes that are involved in the differentiation of cells. That is to say, the development of cells from undifferentiated or stem cells that can evolve into many cell types into the highly differentiated or specialized cells. We found mutations in 2 genes in this pathway, CBFB and RUNX1, neither of which has previously been seen in breast cancers. Mutations in these genes, however, have been seen in leukemias before. The other interesting and potentially even more important finding is a translocation in a gene called AKT3. A translocation means that chromosomes break and rejoin in a new place, and they bring 2 genes that were separate together. Sometimes this provides a functional gene transcript, making a new protein, and these are called a fusion gene and a fusion protein. In this case, the translocation created a new fusion protein called the MAGI3-AKT3 protein. Why this is important is because AKT3 belongs to a class of proteins known as protein kinases. And there are now many drugs available that inhibit protein kinases and treat cancer. In fact, there are drugs that are inhibitors of the AKT kinase that are already in development and have already been introduced into cancer clinical trials. Therefore, our discovery of a translocation in AKT3 provides a potential avenue for treatment for a small fraction of breast cancer patients, and therefore may potentially, if these drug treatments are successful, lead to improved outcomes for such patients. I should note that the cancer in which the AKT3 translocation was discovered was a cancer from a patient in Mexico who was participating in this study.